So welcome back guys. So today we are going to talk about a very interesting topic from the topic genetics. This is including in the topic linkage, and this is about the two phases of linkage, that is one coupling phase, or you can also say this as cis phase, and another one is repulsion, and you can also say this trans phase. So what is those phases, right? If you want they need to know about, then come, let's start. So first of all, before going to the topic, we need to talk about what is linkage and why it is necessary, right? So first of all, let's talk about what is linkage. Linkage is nothing but it is a tendency between two genes to remain together in the same combination in the same chromosome during inheritance for a number of generations. You can relate this thing linkage by simply understanding a example that, let's suppose the two friends, they are best friends among each other, and from class 1 to class 12 they used to sit in the same bench and in the same position, right? Despite the process of inheritance that is occurring to separate them, they remain together for a number of generations. That is the term linkage. That means linkage occurring between two genes those are best friends, and they always used to sit together in the same combination in the same position of the chromosome, right? So now you understand that what linkage is. Linkage is simply the tendency between two genes which are located in the same chromosome, and the distance between them is very less. That is why they always inherited in their same position in the original the combination for a number of generations. That is the idea. That means the main role of crossing over will be inhibited because of this linkage, because this linkage simply enables the plant enables the animal, or you can say the plant to maintain their original combination or parental combination. So first of all, in which the genes used to show the linkage, right? In case of Drosophila, the body color and the nature of the wings controlling genes are linked, and in case of maize, the color of and the shape of the seed are linked. That means linkage can be between any two genes that are located in the same chromosome, and the distance between them will be less. That means linkage play a relationship with distance that will be inversely proportional. Similarly, linkage will always be inversely proportional with recombination frequency. As I have told you that linkage will inhibit the crossing over, right? That means if the linkage is going on between the two heterozygous type genes, so that means two genes are linked together and they are heterozygous. That means one gene is dominant and another one is recessive of other gene. Then if we cross if we undergo a cross with such type of parents with another type of parents, then we should find a increase in recombinant type, as the linkage is shown between a dominant and recessive allele, and coupling and repulsion phase is something of that. Sorry. So linkage have two phases, one is coupling phase and another one is repulsion phase. In case of coupling phase, the dominant alleles of gene that means, suppose let's consider we are talking about a dihybrid cross between two parents. That means that we are considering two parents that are different in different in two characters. That is why we use the term dihybrid, and these two character controlling genes are T and R. If the dominant allele of the genes are located in the same chromosome and recessive allele of them are another chromosome, then the phase will be coupling phase. In that case, if we undergo a cross between parental type, parental dominant type and parental recessive type, then we will have our F1 generation, right? This is something like that. This will be our parental type, and which will give us the F1 generation. And if we perform recessive the test cross with the F1 generation, in normal Mendelian ratio, we will have 1 is to 1 is to 1 is to 1 test cross ratio, right? But in that case, as they are linked, they will show something like that, 7 is to 1 is to 1 is to 7. Not in that case, that is repulsion, that is completely other thing. I will talk about a little time. Hmm. So let's talk about this. So how can you remember that which gene showing a coupling phase and which genes are showing repulsion phase? We can say simply done this by interpreting that in case of the coupling phase, the dominant alleles of the genes are located in the same chromosome and recessive alleles of the gene are located in the same chromosome. That means in coupling phase there is a tendency that parental progeny will be in higher amount rather than recombinant progeny, as there is a higher combination of T, capital T, capital R, capital D, capital R, small t, small r and small t, small r will be in less amount. Therefore, coupling phase shows a higher proportion of parental progeny rather than recombinant progeny. But in case of repulsion phase, dominant allele of one gene is linked with recessive allele of another one and at the same chromosome, as I have mentioned here. So in case of repulsion phase, if we undergo a cross, then the F1 progeny will be test crossed with a recessive parent, then the ratio will be something like that, that is 1 is to 7 is to 7 is to 1, right? Then how this ratio came? This ratio simply wants to tell us that in case of repulsion phase, the recombinant types will be higher than the parental types, right? So if in any MCQ we need to find out that what type of linkage is going on, simply we try to find out which is in higher amount, parental progeny or recombinant progeny. If recombinant progeny is in higher amount, then it will always be repulsion phase, and if parental progeny is in higher amount, then it will always be in coupling phase. Now let's talk about the history of what is a coupling and repulsion phase. So coupling and repulsion phase was first proposed by Bateson and Punnett in 1905, but they could not give a proper explanation about them. Later on, the main hero of linkage, T.H. Morgan, gives the details explanation of this on his experiment of Drosophila. On his experiment of Drosophila, he proved that these two terms, coupling and repulsion, simply are the two aspects of linkage. This is nothing but how the two genes are linked together in the same chromosome. If the dominant genes are located in the same chromosome and they are linked, then we will talk about coupling. And if the dominant allele of one gene is linked with recessive allele of another gene on the same chromosome, then we should talk about what? Repulsion phase. So that is the idea about the coupling phase and repulsion phase. Hope you will like the video. Thank you.